The name of our lab is the Catalyst Activity Lab. This will be done by Jamie Pyatt and Kayla O'Callahan.
The purpose of this experiment is to observe the effects heat has on catalase activity in potato mash. We hypothesize that as you add heat to potato mash, the catalase will become increasingly more inactive. Enzymes are naturally found in many living organisms such as plants, animals, and single-celled units. One type of enzyme found specifically in plant tissues is a catalase. Catalase is not very heat stable and can be destroyed when heated. A practical use of this is to soften unblanched vegetables and get rid of undesirable flavors and odors. The temperature and heat affects the rate at which the catalase will be destroyed. Catalase activity is very high in starchy vegetables, especially potatoes. When the catalase in the potato mash is active, it will react with hydrogen peroxide. The equation is H2O2 decomposes to H2O and O2, which is water and oxygen. Biology is the study of life. Chemistry is the study of matter and the changes it undergoes. Biochemistry is the combination of both of these things. This experiment incorporates biochemistry because there's a chemical reaction when the hydrogen peroxide and the potato mash are combined. It involves biology because the potato and catalase are only found in living things because enzymes are produced in organisms that are living. This relates to Chapter 24 because Section 24.2 is focused on carbohydrates, which is what a potato is, especially starch, which is the active ingredient in the potato that will be tested in this experiment.
In this experiment, the materials you're going to need are filter paper, a strainer, a small bowl, 4 medicine cups, a thermometer, 4 baby medicine spoons, a small pan, a measuring cup, hydrogen peroxide, a blender, a dropper, and a pen, and a small red potato.
Label your medicine cups and baby medicine spoons by numbering numbers 1 through 4 on a paper towel.
Next, add 250 milliliters or 1 cup of water to your blender. After this, cut up your small red skin potato into tiny pieces. Add your cut up potatoes to the blender with your water. Then blend this mixture until smooth.
Once your solution looks like this, add the filter paper to the strainer, put it over the small bowl, and pour your mixture in and wait for it to filter. Once your mixture is completely filtered, pour the solution into a small measuring cup. After this is done, take the initial temperature of the solution. Ours is 27 degrees Celsius. Pour 5 milliliters of the solution into your first medicine cup and set aside for later.
Pour your solution into a pan over the stove and allow it to heat until it reaches 40 degrees Celsius.
Once your solution reaches 40 degrees Celsius, quickly pour it back into the small measuring cup and then pour 5 milliliters into measuring cup number 2, medicine cup number 2. Right. Heat the remaining solution to 45 degrees Celsius and repeat the previous step. Pour this solution into measuring cup, medicine cup number 3. Okay. Repeat the previous steps and heat it this time to 55 degrees. Pour it back into the small measuring cup and then pour it into measuring cup or medicine cup number 4.
Set all 4 medicine cups aside and use your dropper to put 5 milliliters of hydrogen peroxide into each baby medicine spoon.
Rip off 4 pieces of filter paper and dip them into the medicine cups. Once you've done this, make sure you add this to your baby medicine spoon. You can use this to push it down to the bottom. Time how long it takes for the filter paper to rise to the top. As you can see here, this filter paper rose quickly to the top. It took 10 seconds.
Repeat this procedure for the next 3 medicine cups. Could just stop it now. As you can see, this one rose to the top after 20 seconds and there's bubbles occurring. Repeat this step again for medicine cup 3. This one has rose to the top in about 30 seconds. Repeat the procedure one more time for medicine cup 4. It's been 2 minutes and the filter paper has still not risen all the way to the top. In medicine cup 4, the catalyst activity is no longer present.
When you have finished the experiment, dump the waste products down the sink and continue washing with soapy water. Test tube number 1 rose the fastest. Test tube number 2 rose the second fastest, test tube number 3 rose the third fastest, and test tube number 4 did not rise at all. After doing this experiment, we can conclude that our hypothesis was partially correct because while we predicted that the heat would make the catalyst activity decrease, we did not predict that it would stop completely once it reached 55 degrees Celsius.
Thanks for watching!